This is your biology tutor Mpaisoletli. Our topic for today is cloning, a request that was done by one of the students. Now, cloning is the process whereby one tries to copy an organism's DNA. The reason for that is to create an organism that is physically and genetically identical to the original organism, right? So that is the definition of cloning. Now, there are advantages and disadvantages to this, of which we'll tackle later on. But for now, let's look at the process of cloning. So, with the process of cloning, you actually need a donor. Now, in this case, our donor is organism A. Now, from your donor, you want to get a cell, and then after getting this cell, you want to remove the DNA from the cell. That is the only thing you are interested in. And then the nucleus, you want to dispose it. It will be of no use to us, and you get to see why. And then you also need another organism. And from this organism, the only thing that you are interested in is the egg cell. Now, from this egg cell, it will still go through the same process whereby you get the egg cell, and then after getting the egg cell, you want to remove out the nucleus. And then in this case, instead of making use of this nucleus, we are actually disposing it. All right, so we only want to have a nucleus that does not contain a DNA at all. All right, and then now that we have our nucleus without a DNA, and then we have a DNA, we can subject these two to an electrical shock, such that when they are subjected to this electrical shock, they will fuse together. So you have a nucleus that has a DNA. All right, so after having this, your cell will go through a process of cell division that is called mitosis. So it will be growing to a point where this will develop into a zygote. Now that you have a zygote, you want to implant this zygote into a foster parent. All right, so we can call this foster parent to be organism C. And then this organism is just there to carry the zygote, or rather it's just there for the zygote to grow in it to a point where this this organism, or rather this new organism, will be delivered. Now, after delivery, you can see that our organism is actually identical to organism A rather than organism B, or rather than organism C. Now, the reason to that is because we actually used the DNA of organism A. Remember, when we defined cloning, we said that we are making use of a DNA from an original organism. So it's this DNA that we are copying in order to produce new organisms that are physically and genetically identical to the original organism. So this is cloning in short. We made use of the DNA, and then we got to a point where we had a zygote which we implanted in organism C, and then we saw that the individual or rather an offspring that we got was actually identical to organism A. All right, so let's go through the procedures one by one. So like we said, we need to have a donor, and then after having a donor, you want to get a cell from a donor. And then now that we have a cell, we want to remove the DNA from the nucleus. Now, the process whereby one is removing the DNA from a nucleus, that is what we refer to enucleated. Right, now that we have removed the nucleus, or rather now that we have removed the DNA from the nucleus, we are not interested in the nucleus at all. So the only thing that we are interested in at this point is the DNA. All right, the reason for that is we want to copy this organism's DNA to create our new offsprings that are actually identical to organism A. All right, let's move on to the second process. Now, in the second process, you will have another organism which we named to be organism B, or rather donkey B. Now from our donkey B, we'll need an egg cell. Now after getting an egg cell from donkey B, we are still going to remove the DNA from the nucleus. Now the DNA of donkey B will not be in use at all. We don't want to copy this DNA, but rather we want to copy the DNA of organism A. So this DNA is actually disposed. We want to keep its nucleus. The reason for that is we want the DNA of organism A and the nucleus to fuse together by an electrical shock, such that we have a nucleus with our desired DNA together. And then after that, this cell will actually go through cell division, or rather it will go through mitosis, right, whereby it will start growing to a point where we'll have an embryo. So now that we have an embryo, this embryo has to be implanted into a foster parent, right? And then when time is ready, the foster parent will actually deliver to an offspring that is identical to the original organism, which is now donkey A, or rather organism A. So this is how one can make clones, or rather this is the process of cloning. Now there are advantages and disadvantages that are associated with cloning. There are a lot of them, but we'll only go through a few. Starting off with the advantages of cloning, we are told that one is able to save organisms, especially those that are about to get extinct. And then the second advantage is that one can actually obtain more yield. You can obtain more yield in a sense that, say for instance, you were working on a crop, or rather on a certain variety, and then you actually came up with a variety that can resist pests. Now because a crop, or rather a variety that you came up with, cannot be attacked to certain pests, it means that these crops, they will be able to grow properly, such that at the end of the year you'll be able to harvest your potatoes properly. So that is how cloning can actually increase yield. And then moving on to the disadvantages, we are told that there are risk of being infected by the same type of pathogen, or rather by the same type of diseases. Now bear in mind, we actually copied an organism's DNA. So you have an original organism and then you have a copy of it. Now because their DNA are the same, chances that whatever that will actually infect organism A, that will also, we should also expect that in our offspring, or rather in the organism that we have created, because they have the same DNA. So which is also a disadvantage. You cannot have a population, say there's COVID now, because because the original organism got infected by COVID, you get to see that chances that everyone in a country, if say we're cloned, chances that we'll all get COVID is very high, and chances that a lot of us would die, it's also high. So we don't want that. It's good when our DNAs are not the same, because we want to be unique in our own way, such that we can fight diseases, and then survival of the fittest. And then the last disadvantage is that there is fear that we are going against nature. So yes, this is a debate. This is where religion is coming in, and then we have science. So yeah, there are actually a lot of advantages and disadvantages that are associated with cloning. If you know any more advantages that are not listed here, or other disadvantages, please be free to comment down below, and then we'll appreciate that. Thank you so much for your time. If you have any questions, please do comment in the section below. This is your biology tutor Mphayiso Letli. We'll meet again.